In this video, we will talk about the hypothetical development network of the formation of spikes in cells of the ectoderm of the spiny beetle. However, not all cells in the ectoderm have spikes, and some can be smooth. We're given a pathway for the formation of spikes. The T-bar represents the transcription is inhibited, and the arrow represents that transcription is activated. So the S activates the P, which stimulates the I, and the I inhibits the K, and K inhibits the E, and E stimulates the formation of spikes. Now, let's talk about different mutations, starting with the mutation in P. The mutation in P leads to P no longer stimulating I. That means the I is not expressed and can inhibit K. So K is expressed, and therefore K inhibits the E, and E is not expressed. This results in E not being able to produce spikes. So there's no spikes in this mutation. Now, let's look at a mutation in I. I cannot inhibit K, so the K is expressed and it inhibits E. So the E is not expressed, which stimulates no spikes. For the third problem, there's a mutation in E and an I. The mutation in an I would cause it to not inhibit K, and K would not be expressed, and it would inhibit E. But in our case, E is a mutation. So we could have looked at it from the start to conclude that without E, no spikes would be produced because E is furthest down the stream. So sometimes it is easier to work backwards. For the last scenario, there's a mutation in S and a K. Mutation in S would result in failure to stimulate P, and without P, I would not stimulate. And with no I, K would not be inhibited, but K would be present. K would be present, but in this case, there's a mutation in K as well. This means K cannot inhibit E, and this would result in E being present, and E can stimulate the formation of spikes. To conclude, not all ectoderm cells have spikes because they need smooth cells for movement and reproduction, but the spikes do help them from predators.